It's my great pleasure to introduce Oklahoma State Assistant Professor of Microbiology, Dr. Randy Morgenstein. Thank you so much for joining us today.
No problem. Hi, everybody. Thanks for joining. So I thought I would first start with just maybe explaining what is an assistant professor. What's that mean? What do I do, during my day? And then I'll tell you about what I do as research. Okay, so an assistant professor means that I work at a research university. My job is kind of twofold. One, I do research, so I run a lab. In that lab, I have undergraduate students and graduate students. And then I also teach. So you can be an assistant professor and not teach. So people who work at, say, a med school might have they'd still be called an assistant professor. They have a lab, but they most likely aren't teaching because they're not at an undergrad institution. But at Oklahoma State, it's a decently large undergraduate school. So I teach both graduate students and undergrads, and I run a lab. But the majority of my job is to run a lab. So what do I do in that lab? So my lab is interested in kind of the basic understanding of how cells work. So it's really kind of basic biology. We happen to use bacteria as the model organism to study this, right? So you heard I'm a microbiologist, right? So micro means small and biology is living things. So microbiologists study all the small things, the things that we can't see with our eyes, we need a microscope to see. So one of these fundamental questions about cells that we try to answer has to do with the shape of a cell. Hopefully we all kind of maybe have a picture of what a red blood cell looks like, and that would be, I don't know if you guys can see my mouse, but that would be like this cell over here, right? And then there's some white blood cells in this picture as well. No one said neuron, but I'm going to show you a picture of a neuron. Right? And you can see that this neuron is very different than what this blood cell looks like. And epithelial is just like a skin cell, so slightly different than a muscle cell, but closer than the blood cells that people mentioned, or a neuron here. So I'm showing you this is to point out that these different cells in our body all have different shapes, right? And they all have different functions. So a neuron, right, is sending electrical and chemical signals to other neurons, right? It's our brain cells. So the shape of this cell is shaped in such a way to do that. Our skin cells, right, have to form a protective barrier, right? We don't want things coming into our body. So you can see they have these what's called tight junctions, so they're all packed nicely together, make like a monolayer of cells, and then this different blood cells, so a bunch of all the ones that you guys mentioned, right? They all do different things, and they're all different shapes. Okay, so again, this idea of I study fundamental questions of cell biology. So the question is, how do these cells get to be these shapes? But I use bacteria. So if we look at this, oops, sorry. Let me go full screen here. So if we look at this cartoon right here, this is a cartoon of different microorganisms. I think they're all supposed to be bacteria. So one thing I want to point out is the size, right? So we think of microbes, bacteria being very, very tiny, which would be like the ones in the circle here, but there are larger ones, right? So a microbe can get, decently big. And then if I had asked you the same thing to maybe name a bacteria or name the shape of a bacteria, I'm guessing most people, at least because of the exercise that I've heard of E. coli or maybe salmonella, those are common things that contaminate our food, right? So they look like this pink one right here. They're just rod shaped. And then cells can take this basic idea of being a rod and modify it in different ways. So like this one here, right, they took a rod and they bent it. Say this one, it's stretched out and curvy, or this one, right, is a spiral. So it all kind of took that one shape of a rod and modified it in different ways. But then you can see we can have really cool shapes like this star, or I don't know how to describe what this shape would be, and blobs and branch cells, all sorts of different shapes of cells. I'm going to stop sharing there. All right. So what my lab's interested in is using bacteria as a way to start to understand how the cells control their shape. So we use E. coli. It was that simple rod, right? So we're starting with, say, a pretty simple shape, which you might think we should know all about E. coli and how it gets to be its shape, but we don't. There's a whole lot of different questions involving what are the proteins that are necessary to be that shape. So that we have a pretty good idea on. But what those proteins actually do, that's different. And we don't know that. So how do we know what the proteins involved are? Well, you can make mutants in E. coli, and then they're not rod anymore, right? So you can find which proteins are responsible for rod shape by pretty much breaking that protein and looking at the shape and seeing what happens. Now, figuring out why the shape got messed up, right? What is that protein doing? That's a more difficult question. So that's one of the areas my lab is working on, is taking these known genes involved in cell shape and asking what they do. One of the things we do is 3D microscopy. So grab my little prop over here. So here's our cell, right? It's a three-dimensional object. It's very, very tiny, but it's still a three-dimensional object. So when most people take an image, they kind of, with a microscope, they're just taking like a flat picture of the cell. So a two-dimensional image, kind of just like a photograph. So what we developed is a three-dimensional way of imaging the bacteria. So what we'll do is we'll take a picture at the top and then just move down through the cell and take a whole bunch of different stacks, which I'll show you in a second. And then we can take those and put them all together and get a pretty accurate representation of what this cell looks like in 3 dimensions. So over here, what you're seeing is we have a membrane stain of our cell, and it's doing that, taking a picture from the top and going all the way through to the bottom, right? So when we start on one side, it's out of focus, and we come in, and you see it's in focus, and then we're going down, and it's out of focus again. So then we can, using a computer, we take all of these images, and we can get this cell out of it. So this now is a 3D representation of all of these images. And then what we can do from there is we can have a protein of interest that is fluorescent. So that's what you see down here. So we're doing the same, taking that stack of images, and now we can map this protein onto our three-dimensional cell. So you can see the heat map here. So these dark red areas is where this protein is. So like if we wait till this comes back into focus, right over here, that would be like this area over here. So what we can do with that now is we have a better understanding of the curvatures of the cell and really where proteins localize inside of the cell instead of just looking at one plane. So I said like we are trying to figure out what those proteins that give cell shape do, and then we also use cell shape kind of as a tool to understand other things. So many antibiotics target the cell wall of a bacteria, and the cell wall is what gives the cell its shape. So by studying shape, we're studying the cell wall, which is a huge target for antibiotics. So it kind of opens up ways to understand how the cell becomes resistant to these antibiotics. So we have some different work looking at antibiotic resistance and some kind of new mechanisms that we've discovered on that front. The typical day kind of comes back to what I was telling you about what an assistant professor is. Right. So sometimes I'm teaching. So you guys all know what that looks like. When I'm not teaching, surprisingly, I'm not actually in the lab at this point in my career as much as you might think. Right. So up until now, all I've done is lab work. So graduate from undergrad, go to grad school. In grad school, you take some classes, and you work in the lab like full time. At some point during grad school, you stop taking classes, and all you do is lab work. Then you do what's called a postdoc. So that's kind of a research position in between your graduate school, so getting your PhD and, say, becoming the professor. You do a postdoc. And a postdoc is just research, right? So pretty much grad school is research, postdoc is research, and then you become a professor, and now you go from doing research to mentoring. So my day doesn't involve doing research at the bench anymore. I do a little bit, and every professor is different. Some people probably never go in the lab, and some people really try to make sure they can still get in there. But either way, it's way less than you did throughout the rest of your career up to this point. So now I'm mentoring students. So I meet with the undergrads and the grad students who are in my lab, talk about their projects, what they're doing, if things worked, things didn't work, why or why not. Uh, One of my main jobs is actually to get money to fund the lab, so pay salaries, buy equipment, all that kind of stuff. So there's a lot of writing grants. So that's kind of going to be cyclical, right? If you have a bunch of grants, you don't need to write anymore. As your grant starts to, they're normally 3 to 5 years. So right as your grant starts to end, you go back, you kind of get back into grant writing because you have to get the next grant. Right now, we're working on publishing a couple papers, so there's a lot of my time spent on writing papers, editing papers, working on the figures, that kind of stuff. And then there's um, community service, I guess, type things. So professors review papers that other people write. So like we work for free for journals to review the papers to make sure, right? Hopefully you guys know, right? You want to trust peer-reviewed science, right? It means that other scientists have read it and been like, yes, this looks good. The science was done well. There's the proper controls. This is something that we can believe versus with COVID is kind of a good example. There's a lot of stuff when COVID first became a big deal that was like all these studies that hadn't been peer reviewed yet, but the news was picking up on them. So right before it's peer reviewed, no, it's just a guy telling you, or a woman telling you something, and there's been no confirmation that it was done in the correct way. So there's a certain part of my day that I spent reviewing papers, and that's kind of cyclical too. So journals will send them to you and ask you if you have time to do it. So sometimes you're doing a bunch, sometimes you might not be doing any for a while. So that's kind of what my day is like. So what inspired me to go into science? I don't really have a good story for that. It's not going to be enlightening to you, but I guess in high school I had a good biology teacher. So we didn't have, I don't know if you guys do or not, but we didn't have like an AP bio class, but I had a biology professor who kind of did a after school thing for people who like wanted to take the AP bio exams. So he like kind of taught an after school study class to do that. So me and a few other people did it, and I did well, and I was like, oh, this is pretty cool. So, and my father was a doctor, so I was kind of always around sciencey stuff. He was an infectious disease doctor. Now I do microbiology. So kind of just was always around science. Was pretty good at it. I guess had the right kind of teachers in high school to kind of push, and when you find something you're good at and enjoy, it's a good thing to try to make a career out of. So I just kept kind of doing it, and I still enjoy it. So it's worked out pretty well for me on that front. Microbiology is actually somewhat random that I ended up in microbiology. When I went to Berkeley for undergraduate education. So when you're filling out the application, you can fill out a major, and at Berkeley, the microbiology degree was a BS degree, so Bachelor of Science, and the like molecular and cell biology was a BA degree. So I was like, well, I want the BS. So I just clicked the microbiology box, and it worked out pretty well. I enjoyed it. I'm happy I ended up there, but it was kind of serendipitous and slight accident. Any tips, advice, or hacks for finding success in school? So I will say, both as a student and now as a teacher, that high school may be easy for you, high school may be difficult for you, but either way, college is probably going to be more difficult. So try to learn good study habits now. I know for me this was a problem, and many of my students who say they did well in high school, maybe they didn't study very hard or they didn't try very hard, and then they get to college and it's a different story, and it might take a year or two to pick up those study habits. So if you can work on it now when it's easy for you, it will definitely be useful for you going if you go to college for the future. So I will say do that, and also just kind of remember everyone studies differently. Right? Some people like to write stuff down. Some people like to listen and watch things. So you have to. That's part of it is like figuring out what works for you and doing that. Don't listen necessarily to what someone else tells you is the right way to do something, because there's no right way, right? Whatever works for you is what you want to do. right? Some people, I'm just going to read the book and that's all I need to do. Some people, I'm going to read the book and take notes because if I write it down, it helps me remember. So you have to figure out what's going to work for you. See a question here in the chat, so I'll answer that. Yeah, so greed is a fun, interesting term. Yeah, so E. coli, right, exists in nature, exists in you, right? So E. coli is like a gut microbe. It's in everybody's stomach. Um, it's out there in nature. We, because like again, we're asking these fundamental kind of questions. We use what's called a lab strain of E. coli. So we're not going out into nature and isolating variants and doing stuff with, either stomach E. coli or soil or animal E. coli. There's many different lab strains of E. coli. I don't know how many. There's a few different ones that most people use. So we use one of those. And so is it, as natural or original as like if you isolated one from your stomach? So the answer there is no. It's definitely been evolved to grow in a lab over 50 years or more. So that could be a problem. There are people who, I don't know E. coli as much, but other kind of model organisms, there are people who will try to use wild isolates to get a better idea of things that haven't been grown in the lab for forever. Like how do they work? But we use almost exclusively these lab strains. So one thing that's great with working with bacteria is they grow very, very fast. So E. coli doubles every 20 minutes, right? So we can grow culture overnight, and there'll be billions of cells the next day, and we can take those cells and do whatever we want, that day with them. We come up with an experiment. Oh, this would be a great idea. I can do it tomorrow. If you work with something that grows slower, you have to be pros and cons, but you maybe have to plan a little bit better because you can't be like, oh, this is a great idea. I'm going to do it tomorrow. Well, you can do it next week because it takes a while for your cells to grow. So one of the nice benefits of working with E. coli and most bacteria, there are slow-growing bacteria, but many bacteria grow very fast. So you can come up with an idea, maybe do it that day, do it the next day. So those are good things. So that kind of also answers the how do we make sure everything remains constant throughout the testing, right? So we're using one strain. But that is kind of a good point, right? So there are things called controls, right? So if we're doing something with a mutant, we always have our normal called wild type strain, so we can compare them. And then if you have a mutant, I'm going to throw you some terms out there that you might not have heard of, and I'll explain it. You want to do something called complementation. So what that means is I made a mutant, so I deleted a gene from my cells, and now I'm going to put that gene back into the cells. Right? So I deleted this gene, and my E. coli went from a rod, to a round cell. And now to make sure that they became round because of what I deleted, I'm going to put that gene back in and make sure it's rod again. So there's all sorts of kind of controls like that to make sure what you're studying is what you think is what you're studying, and not something else weird happened and what you think you're studying is wrong. Okay, lots of questions coming. So I'm going to go with this one, I think, from a student. Maybe, how do you learn? What method of learning is best for that person? Yeah, I think that's just trial and error, right? You're going to have to, if you have your textbook, I don't know if you guys taking biology or chemistry or something. Chemistry is normally pretty hard for people. Hard for me, I know that. I can show you my college chemistry books. I literally highlighted like the entire book. Well, what use is that when you go back to studying the entire book is highlighted? So that, not the best method, but some people, they read things and they absorb. So I think you really just have to kind of figure out. That's why I'm saying if you start now and things are a little bit more easy, maybe you figure out, okay, just reading is not really enough, but if I take notes while I read, so like the act of writing, for example, helps people. Other people are more auditory, so maybe if you read the book out loud, or lectures themselves are super helpful because you hear it. So yeah, I unfortunately can't tell you how to figure it out other than a little trial and error and just kind of seeing what works for you. Yes, I'm very happy with the choice of work that I do. One of the benefits of my job is, so I dressed up for you guys today, so you can see I don't have to, wear a suit or anything like that. And I'm pretty much my boss, my own boss. I have, there's administrative structure. So like the chair, I'm in a department of microbiology, so there's the chair of the department who would be my direct boss, but on the day-to-day aspects of my job, I don't report to anybody. I don't report to the chair, so I can come and go as I please, take vacation when I want, all of those kind of personal things is kind of a nice advantage of being in this position. Research-wise, being a professor, you get to work on whatever you want. So kind of the normal breakdown is, academia, become a professor type of thing, or go into biotech. So biotech, for the most part, you're going to make more money, but what you're going to research is going to be much more limited. Right? You're going to do what the company wants, something that's going to end up making money, right? That's the goal of a biotech company, is to make money. So you may be really interested in cell shape, but you're not going to be doing that in a biotech company because they don't necessarily care. So, right, as you go through school, at some point I thought I wanted to go into biotech. That was my original plan. And then I just kind of was like, I really like deciding what I do. So for me, it was more important to have that control over what I study than, make the extra money or things like that. I would say I don't know which one has more job security. Maybe biotech. So I'm an assistant professor, so that means I don't have what's called tenure yet. So before you get tenure, you can kind of be fired and try to have to find another job. Then I'm be coming up for tenure very shortly. So once you get tenure, then you have job security because that's pretty much you're there for the rest of your life if you want. It's very difficult to fire somebody with tenure. So kind of early in your career, you have less job security, and then later in your career, you have more job security. And I presume in biotech, if you're doing a good job, I don't think big biotech companies going out of business. But if you work at a startup or something like that, right, less job security because the startup could not go out of business. Something about the 3D pictures. Yeah, so the process of the 3D pictures fail all the time. There's a lot of quality control steps that we go through. So if you guys have a chance later, after we post that video, you can kind of see how it's kind of similar to what I showed you on my PowerPoint. We take those stacks, and then the computer tries to assemble it. Sometimes it just never assembles, and that still obviously gets thrown away. Sometimes it does assemble, but it has like a weird blob coming off of it because something messed up. So those cells get thrown away. Sometimes there's a hole in it, right? So again, we had our cell and we took all these pictures, but then sometimes it didn't do something right and we end up with a hole in it. So, we try to, when we take images, of 300 cells so that maybe we have 100 cells at the end that actually worked, and then we can work with. Hopefully that answered your question there. But it's pretty much like any experiment you do, things might not work. So that's also why we have those controls. And you just you minimize it. You do the experiments a bunch of times so as you get better at it too. Hopefully that will limit things not working. It's going to be very specific, right? If you become a microbiologist versus a eukaryotic person, or biochemist versus a geneticist, it's a lot of school involved to get to become a professor. So you will learn your vocabulary that you need for your field as you go through. Yeah. So one of the things with bacteria is that they grow very quickly. So contamination is an issue, but it's not. If you work with something that grows slow, you have an issue of it gets contaminated with bacteria that grow quickly. So thankfully, we're already working with the quick growing stuff. We wear gloves. We have Bunsen burners, right, with fire, so that helps to keep things sterile. we spray things with ethanol to sterilize things before we use them, things like that. Um, and it is a big worry about letting things into nature, right? So there's a lot of work that people don't do, like we don't try, like I think you need special permission to make things more pathogenic, for instance, right? It's not something we want to do because you don't want to get it to escape out into the world. So when we're done with everything, we do what's called autoclaving. So we put it in a big like pressure oven to kill it. You can, or we bleach it or something like that. So we make sure that everything is killed before it has a chance to get out there. Um, there's lots of regulations at the federal and state level, and then the university has a whole like department for biosafety and stuff where you have protocols that they have to approve to make sure everything you're doing right conforms to the rules and that you don't have the risk of things escaping. I know one thing is like all our windows in the lab have to have screens on it just so like a fly couldn't fly into the lab and land on your plate and pick up bacteria and then like fly off the window or something like that. So it's definitely a worry, but we don't just pour things down the drain or anything like that. You know, if things are leaving on us, that's probably a bigger problem. But we're washing our hands, trying to keep everything sterile, so that shouldn't be a problem. Home lab experiments. You should be able to get at least probably isopropanol or something like that. I think at a drugstore that you can use to sterilize certain things. Trying to think of like, to be honest, if you could like open a box of Q-tips and we're going to take a swab, obviously it's not like sterile, but it's probably, I've not done this, but I bet you it's pretty decently sterile. But this can kind of come back. So for your home experiments, I was mentioning doing controls. So say, I don't know what home experiment you had in mind, but let's say you had bought some petri dishes and you wanted to like see which toilet in your house had the most bacteria on it. Right. So if you're like, okay, I'm going to use this Q-tip from our bathroom and I'm going to go rub it on different toilets. So you can take a Q-tip and not rub it on anything, right? And say put it on your plate and then see what grows from that, right? So that's kind of what would be your negative control, right? So nothing should grow in that one. So I will tell you how sterile your Q-tip was to start, and then you can kind of base your other experiments off of that. But depending on what you're doing, I think, I'm not going to say that you should steal your parents' vodka to sterilize things, but I think you can get from the drugstore isopropanol and things like that. So that's the alcohol that can sterilize things. You can probably use a candle and put something through the wick if you want to heat it to sterilize it. Don't burn yourselves on do these things with parental consent and watching you to make sure you don't burn the house down. But so you could probably do pretty much the same things we do in the lab, except for the autoclave part. But you could do many of the same things that we would do in the lab in your house. Process of proofreading papers. So there's kind of two things, right? So one, the basic stuff that you guys are learning in school, right? Grammar. Be surprised how many scientists just forget about English and don't write very clearly, right? You can have the coolest experiment, the most impactful result, but if you can't relay that information to other people, be it the general public or other scientists for publication in a journal versus say like a newspaper article or a blog or something like that, it doesn't matter. So that's kind of one, is writing things with proper grammar, proper commas and all that kind of stuff so that it actually makes sense. And then we're reading things for the science. So did they do the experiments correctly? Why did they do the experiments the way they did it? Going back to those controls, right? Did they have the proper controls that they do things in enough? We do things in replicates, or people joke it's called research because you redo it all the time, right? So we do things over and over again. You don't just get a result once and ta-da! You redo that experiment multiple times to make sure that the result you get is consistent. For us, let's say with cell-shaped things, each time we do it, we want to make sure we have at least like 100 cells per strain or whatever we're comparing, and then we'll do it again the next day around another 100 cells. So are there enough replicates? Are there enough what's the term I want to use? Are there enough things in that replicate? So in my example, like, are there enough cells? If we just imaged 5 cells and said, hey, look at what the shape of this population is, 5 cells, it's not particularly not necessarily representative of, the whole population. So are there enough cells? I can't think of the actual term I want to use, but hopefully that gets the point across. Things like that is what we're looking for. So I don't know if you meant when I was reviewing, if I'm reviewing a paper for a journal or I'm writing a paper or reviewing like my grad student's paper before we submit it, but it's grammar. Did we explain what we did clearly? Did we do everything correctly? Things like that. Jump back up to a question that was, what is the most important part of my job? And that might be depending on on who you talk to. My grad students might think the most important part of my job is, mentoring them and helping them along on their careers. Maybe the students whose class I'm teaching think the most important part of my job is teaching. Unfortunately, the most important part of my job is probably writing grants and making sure the lab is running. But to me, the most rewarding part is the mentoring students, and also this kind of comes back to some other questions. But one of the other kind of cool thing about my job is, I'm the first or second person in the world, say, to know something, right? We do experiments and we discover things. So I'll say I'm the second person now because the grad student did it, so they know the result before they tell me. But so I'm in the very small group of people who know something before anyone else, right, in the history of the world has ever known something because we discovered it. So that's also something that's pretty cool about my job. So this most recent question. So everything I know about microbiology has changed my perspective on the world. I'm actually going to say I think most microbiologists are way less worried about things than other people. I am definitely a proponent of the 5-second rule. So less so now that I have a dog because they're licking the floor all over the place. But before that, I definitely am less concerned about things, I think, than other people are. So this kind of question about publications and popular social media and stuff like that. Yeah, so I think we've seen this really well with COVID and how in the very beginning people said certain things, say mask wearing, at the beginning when there was very low PPE around and it was like, don't wear a mask, kind of save it for the hospital workers. And then later became everyone should wear a mask. I feel many people think, oh, you're just changing things as you go, and you don't know what you're talking about. We're not going to listen. We're not going to listen to you. Scientists don't know anything. But really, right? Or even how 6 feet, 12 feet, how much social distancing do we need? Right? All these things, as views change, that's what science is, right? We do experiments. There's some hypothesis. Okay, we think 12. We'll go with, I'm making up numbers for social distancing. 12 feet seems to be good. And then we do more experiments and, it could be 6 feet. And then maybe more people do experiments like, there was some problem with what they did. Let's go back, a little bit to 10 feet, right? So I think things being, especially back when I said like all that stuff that wasn't peer reviewed yet, but the news would pick up on it and start talking about it as if it was, the new dogma, like this is from God himself told us this information. That's kind of a problem. And, it'd be good if media waited until someone else had read it and, it doesn't have to be published yet, but at least maybe there's many steps between when you submit a paper and when it gets published. But at least it had been peer reviewed and there was some idea of, okay, yes, it seems like the methods they use are good. There's maybe some more experiments we need to do or another analysis to do on the data, but for the most part, what they said is true. So talking about things too early can be bad. I think we're all familiar with clickbait and change titles of things. So that's, I think, a big deal when media on Facebook or anywhere makes some really outlandish title of something as clickbait. So you click on it, but then most people don't click on it. They just see the title and move on. But now in their head, whatever that crazy title is true. But if you read the article, you'd see, right, it was not true. So that's a big problem. And I think responsibility in the media, the way they represent science, is really important. And I don't think they care too much about it, right? It's more about clicks and views and stuff than it is about getting the accurate information across. So that can be, I think, a pretty big problem.
Are there any microbiologists that are working on solutions for climate change?
Yeah, so let's say there are so our department is pretty fun because we have a nice mix of like people who are working on pathogenesis, people who are working on environmental microbiology, and people kind of me who's in between all this stuff. We'll just call me a cell biologist. So some of our environmental microbiologists uh, do things like bioremediation. So I don't know if you guys have heard of bioremediation before, but that's using organisms to clean up the environment. So it's not necessarily directly related to global warming, but I'm in Oklahoma, right, so we have a lot of oil fields and fracking and stuff like that. So gas as well. So fracking and those types of things happen here, and so the water. This is, I think, for fracking and oil wells. I'm not an expert on this, but there's a lot of water used to flush things, and that water is super contaminated with stuff. Yes, plastic-eating bacteria is an example of bioremediation. Uh, Oil wells and stuff, flush it with water, and that water is super contaminated. So we have people in our department who are working on isolating organisms that could help to eat some of those compounds that would be in this contaminated water. Coming back to global warming, I don't know if there's no one in our department is working on it, but we have maybe someone at the university. But a big source, or I don't want to say big source, a source, right, of global warming is methane, and a big source of methane are cows, burping and other gas released from a cow, and the methane that the cows release actually comes from microbes in their guts. So there are archaea. So they're not bacteria. They're another microscopic organism. They're not eukaryotes either. This is the third branch of life. Some of you maybe learned about that in the kind of tree of life there in the biology class. So there are archaea that make methane. So they help the cow digest like the grasses that they eat. And then these archaea make methane, and then the cow, burps out the methane. So there is, depending on how much cows are causing global warming, so there is a connection there with microbiology, as the methane does come from microbes. But yeah, our department itself, we're not too interested in. I don't want to say we're not interested in it, right? Everyone's interested in it in the sense that it harms or it influences everybody, but we're not doing direct research on global warming per se. That's a good question. So could someone engineer some kind of new microbiome for a cow to reduce methane? Yeah, maybe. Because really the key is you need to help the cow digest the grass it eats. So one thing that we can do in biology is take genes from one organism and move it to another organism. So maybe someone could take some of the genes involved in breaking down the grasses and move it into an organism that wouldn't make methane. Maybe that could work. It'd be tricky. I don't think we could go replace the microbiome of every cow on the planet, but maybe every new cow you can try to do something with. So maybe over time we can kind of transition from what we have now to a better thing. I don't know, to be honest, if anyone's working on that, but it's a nice idea. So thank you for that comment. Yeah.
Well, that just about wraps up our question and answer. But thank you so much, Dr. Morgenstein, and we're so grateful for your time.